Hi there, everyone. Thank you for everyone who is logging on right now. We're going to take a moment just as people are popping into this event tonight. Thank you so much for logging on to Zoom to attend tonight's Popping the Science Bubble. All right. And it appears we are live on Facebook now. Thank you so much to those who are tuning in on Facebook Live. All right. All right, so we'll just go ahead and get started here. Just wanted to thank everyone for tuning in tonight. Welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they find really interesting. The organizers are 3 graduate students at UC Berkeley, Jenna, Madison, and Oksana. And Popping the Science Bubble is a Berkeley Public Library favorite event. It's a partnership we are just so excited about. If you're interested in checking out some of the past seminars that we've done, you can visit the Popping the Science Bubble website, or you can check out their YouTube videos where this talk tonight will be uploaded. And you can also sign up for a listserv that will keep you in the loop for future seminars. Thank you so much for logging in, and I'm going to pass it off to the Science Bubble team to introduce tonight's speaker.
Thank you, Kelsey, for the intro. So we just want to start by saying that we encourage you to ask questions during the course of the talks tonight using either the Zoom Q&A button or the comments on Facebook. And if you're interested in learning more about Popping the Science Bubble seminars, and if you want to see some of the past presentations, you can visit our website and social media, and you can also stay connected with us through our listserv. And Jenna will now introduce our first speaker.
Okay, so we'd like to welcome Maya. Maya grew up in Alabama and completed her undergraduate degree at Washington University in St. Louis before joining the Integrative Biology department at Cal. She studied many different things, including forest fires and extreme microbes, before finding her passion for ancient oceans. When not doing research, Maya enjoys camping, baking, and playing music. And let's listen to Maya speak.
Okay, thank you for that introduction, Jenna. And can you give me a thumbs up if you can see my screen? Awesome. So hi, everyone, and thanks for being here tonight. I'm really excited to talk to you about moss animals, the secret lives of overlooked ocean creatures. I study weird little known animals, and I'm excited to tell you this evening about why they captivate me, and hopefully you'll come to appreciate them and value them too. But before I can explain what I research, I have to explain where I research. I have long been captivated by beautiful natural places like this. And I am motivated to read all about them and research them because the more I understand, the better I can appreciate and care for them. But what's so interesting about coastal environments like the one in this picture? What draws both herons and humans to these places? While the space between high and low tide can look pretty barren and wave battered, if you look closely, you'll actually find a lot of strange things. For example, you might find sea stars like this. The very first time I visited a tide pool, I was familiar with classic sea stars like this, but I had a hard time even believing that these two were real living animals, but they are. This is a basket star and this is a slime star. And believe it or not, all of these sea stars are voracious carnivores that are really like the lions or wolves of the intertidal. You might also find sea urchins and cucumbers. Now, these three don't look anything alike, but their species are actually very close cousins. They eat sandy sediment from the ocean floor and digest the nutrients within it. And they don't have eyes like we do, but somehow, and scientists don't know how, they can see their surroundings and run from predators like the sea star. And if you were to look at the intertidal very closely, you might notice this camouflaged mollusk called a chitin. And if you were strong enough to pry it off the rock, this is what its body looks like underneath. And it uses this mouth to scrape algae off the rocks. So you can get a sense from the photos I've shown you that it can be hard to even realize intertidal animals are animals. They have tentacles instead of the arms and legs we're used to, and they have no heads or faces in the sense we're used to recognizing. And I like to think I study the weirdest intertidal animal of them all, the bryozoa. Bryozoa were named bryozoa by the first Western scientists to catalog them because it's Greek for moss animal. And if you were to come across one of these, my guess is you would also be likely to think it's a moss or a fungus instead of an animal, but it is. And I hope to convince you in the next few minutes that these very odd animals are very valuable and interesting. First of all, they don't all look like that. They actually come in a diverse variety of shapes and colors. And I'd like you all to take just a moment and look at these photos and try to think what all of these different bryozoa have in common. So maybe you notice that most of them have this repeated lace-like pattern of circles or boxes. And if we take a closer look at one of them, we can see that lace-like pattern better. And this is the crazy thing about bryozoa that I love. This whole thing is actually one bryozoa colony, and each box you see is one individual bryozoan animal. And that's because they're colonial organisms, which means a bunch of individuals live together in a connected, growing colony, which is like a shared skeleton. And each of these little tentacles you see sticking up from each box, that's their mouth. So let me back up and let's try to understand what a colonial organism is and why it might evolve. So take a moment and think to yourself, what's an example you know of an animal that lives in groups? And if you'd like to, type an idea in the chat or comments. So far, one suggestion we've gotten are corals and ants, both great examples that I'll talk about. Monkeys, that's another great example. So I'll just wait a little longer if anyone else wants to add their ideas. There are no wrong answers to this. Humans, bees, yes, these are all fantastic examples. So I would say that all of these examples fit into three categories I'll explain: social, eusocial, and colonial. So social animals includes monkeys, which was one great example, and also humans. These live in relatively small groups, usually of closely related individuals. And usually they live together for coordinated hunting or for safety. So two examples you're probably familiar with are lions and dolphins. But an intertidal example you probably haven't heard of are these feather duster worms. So each of these worm tubes is about 1 foot long. They're really big. And they choose to live in groups. And when I walked up to this colony of feather duster worms to take this photo, one of them sensed my presence and warned all of the others, and then all of the worms disappeared back into their tubes. So you can see, just like lions and dolphins, part of the reason these worms live together is for safety. Then we have eusocial animals, like bees and ants, which were both great examples people provided, where they live in huge groups, and every individual has a specialized role it plays. And usually there's only one or a few queens that are the only reproductive individuals. And one example of this you're probably less familiar with are naked mole rats, which actually live underground with one dominant queen. And then we have the colonial animals.
Oh, hey, Maya, just a quick question about the previous slide. Someone was interested about what the bees are doing, where they're going.
Good question. I did not take this picture myself, but I believe this is a beehive being used for honey. So this is one of the entrance holes someone has carved for the bees to get inside their box.
Thank you.
So then we have colonial animals, which is like the highest level of organization. And this is where all of the individuals actually are connected by a skeleton or tissue that they all share. So corals like these are probably the most familiar example. And then one you might not have heard of are tunicates, like this. And then we have my study animals, the bryozoa, here in the middle. So all three of these groups evolved this colonial lifestyle separately, and it can be advantageous because they can share food and resources and specialize in specific jobs, just like bees in a beehive. They can also grow bigger than any individual bryozoa could, which allows them to compete for food and fend off predators. But what do these eat, and what does one individual bryozoa look like? Well, if we zoom in on one colony, you can see each individual box in the skeleton, which, like I said, is one individual, but it looks empty. But it's not. They do have organs and tissues inside. They're just so small and transparent that they're almost invisible. But if we look at this diagram over here, you can see that inside they have this feeding tentacle that they can put out when they want to eat and pull in. And they just eat plankton that are floating by in the water. And some of them can grow extra specialized appendages for specific jobs they serve to the colony, but this is pretty much it. They're basically just a box with a feeding tentacle. And once a bryozoa settles and begins to grow, it doesn't move. So it actually seems like a pretty plant-like lifestyle where they pick a spot, they grow in that spot, and they just passively feed what's around them. And so it seems like an example of an animal acting like a plant. And there are a few other intertidal animals with this strange lifestyle, including this sponge, this barnacle, and this sea anemone. And animals that have this lifestyle are called sessile, which is a word for things that don't move, suspension feeders. And a suspension feeder is something that just eats things that are suspended in the water. And this is a lifestyle pretty much only done by marine animals and no land animals that we're familiar with. And it's just one example of the boundary we think there is between plants and animals kind of breaking down in an interesting way. Another cool example I wanted to be sure to include are these sea slugs. Yes, these are all slugs that actually photosynthesize and pull energy from the sun just like a plant to survive. As you can see, they even mimic the structure of a leaf. And these coincidentally are also bryozoan predators. So the line between plants and animals is more at a cellular level, and we can't always make that differentiation just by looking at them, because not all animals walk around and not all animals necessarily hunt for food. Now there's one last cool thing I want to drive home about bryozoa, which is that of all of the cool intertidal animals we've talked about, they have the best fossils. So all of these are fossils in Cal's Museum of Paleontology, where I often work. And as you can still see that lace-like pattern of the bryozoa in the rock. And a bryozoan forms a fossil when it dies and then is buried under a lot of pressure in the sandy sediment on the ocean floor. And we can learn everything we could about the living ones from the fossil ones, so we can compare how they've changed over millions of years. And bryozoa have actually been around for 480 million years. And so let's take a minute and try to compare that to animals we're more familiar with. Just like we did before, if anyone wants to type a suggestion in the chat or comments, what's the oldest type of animal you can think of?
And while we're waiting for people to think about that question, kind of a related question in the chat and one I had is, could you give an estimate of the size of like that bryozoa slug predator and then like one individual box of the bryozoa?
Yeah, great question. So one of those slugs is maybe 2 to 3 inches long. And an individual bryozoa is actually microscopic. So up to 1 millimeter, usually less. But the colonies can be like feet long. So the colony's body size is pretty substantial, and they're big enough not to all be eaten by one of those sea slugs, but each individual bryozoa couldn't make it on its own.
Okay, really cool. And it looks like there's some answers in the chat if you want to look at them, or if you want us to read them.
So ammonites, that's a great one. I'll definitely talk about. Sponges, another great one, and early humans. So let's visualize.
We also have some suggestions on Facebook. An alligator.
Oh, alligator, that's a good one. So I'll talk about all of these using this chart. So here we're picturing the history of the Earth as a spiral, where the Earth forms down at the bottom here 4.5 billion years ago, and then we spiral up to the present day. So here we are in the present day, humans. And then moving back, here 65 million years ago, we get the last ammonites, which was one person's suggestion. They were around, or rather they ended at the end of the time of the dinosaurs. So here's the time of the dinosaurs. You can see a bunch of them pictured here. And alligators are also reptiles descended from one branch of dinosaurs. So they originated around here and then persisted to the present day. And then as we move back, here we get to when the first dinosaurs were alive. And then here we get pre-dinosaur. And then back here, you see this little fish climbing onto land. This is when the first animals moved onto land. So as you can see, through most of the history of life, all of life was actually in the oceans. So then as we continue to move backwards, here we have kind of an early squid. Moving back, here we have the first mollusks, trilobites, which are the ancestors of all modern insects. And around this early period where you kind of get the first mollusks and arthropods, this is when bryozoa are from. And they've persisted in basically the same form all the way back, from all the way back here to the present day, which is a really long time for the same colonial lifestyle to persist. And sponges, coral, tunicates, all of those have been around for the same length of time, but bryozoa happen to fossilize a lot better, so we can make comparisons of how they've changed over millions of years over a much longer time scale. And where do we find bryozoa fossils? Well, you can find them in rocks that formed underwater all over the world, including right next to where you would find living bryozoa. So this is a photo I took at a beach just south of San Francisco. And I was looking for living bryozoa in the water when, looking in the rock face behind me, I found all of these marine fossils. These are maybe 20 million years old, still pretty old. And as you can see, there's not just bryozoa, but a whole diversity of intertidal animal fossils that are right next to where you find the living individuals. And so this allows us to make comparisons about how they've changed over time in the same locality or in the same part of the world. And this is something that you all can do too. There are tons of places to find fossils and intertidal animals at tide pools all around the San Francisco Bay. And if you're tuning in from elsewhere, there are actually bryozoa in every body of water, marine and fresh, all over the world pretty much. They're super versatile, so you can definitely find some near you. Just be sure to check the tide charts and try to go during a low tide during your visit. I also checked, and there are lots of interesting books about intertidal animals in the Berkeley Public Library. And a lot of the photos I've featured today are from iNaturalist, which is a great app if you want to learn to identify some of these things, because nowadays it can actually identify or give you the name of the species just from a photo you take. So those are all great ways to keep exploring this. And with that, I'm happy to take questions.
Awesome. Thank you for this great talk, Maya. I was definitely intrigued by like basically all these animals I've never seen or heard about before. So super cool. And let's see if we have any questions. I know I do. Okay, so yeah, we do have a question for you. Is it known how individual marine animals organize themselves into a skeletal colony?
Great question. So when the colony starts, there's just one individual, and then that individual buds off and gives rise to all of the other ones in the colony. So it's a lot like a plant. If maybe you're familiar with succulents, how you can start with one leaf and plant that one leaf and it will grow the rest of the plant. This is kind of like that, except you start with one individual that buds off to produce the rest of the colony. Hopefully that answers the question.
Yeah, I think it does. Thank you. And it looks like we have a Facebook chat question. So you talked about the worms that warned each other when you were approaching. Do you know how they're able to communicate to warn each other?
Absolutely no idea. And I doubt anybody knows, because there's more unknowns than knowns about how these strange animals sense their surroundings. Like I also said about the sea urchins and sea cucumbers, we know experimentally that they can literally see around them, but they don't have eyes. So there's some way to see without eyes, and no one knows how it works. And it's just kind of mind-blowing and freaky.
Yeah, that is really strange to think about, that they could be watching you, but you don't know how they're doing it.
Yeah.
Yeah. Okay. Let's see. Yeah, and if people are thinking about questions, I'll ask my question in the meantime. So I guess now the bryozoa remind me a little bit of coral, and I feel like in the news we've heard about coral, being impacted by like rising ocean level temperatures or acidification. Have you seen bryozoa also being impacted by like recent changes with the ocean?
That's a great question. So like coral, bryozoa are making their skeletons out of calcite, which is a lot like limestone. And if you're familiar with limestone, you know that it dissolves in acid. So as the ocean gets more acidic, it literally just dissolves their skeleton, which directly kills them. So we know, because their skeletons are similar to coral, that they're going to suffer the same effects. However, hardly anybody studies bryozoa. So no one's actually tabulating the diversity and keeping track of whether they're disappearing or not. So no one knows how well they're doing.
Oh man, okay. So it sounds like this is kind of a really open field still in terms of studying bryozoa then?
Yeah.
Okay. Oh, we have a couple of questions from the audience. Oh, a very interesting one. Are you ever tempted to taste mosses that you collect?
That's a great question. definitely would not, because a lot of these intertidal animals produce toxins and poisons to deter other animals from eating them, including bryozoa. They have really strange chemical properties. There's actually one cancer drug that's made from bryozoa because of its potency to kill cells, basically. So if I ate one, I'd probably die.
Okay. So PSA for people, don't eat moss that you find at the beaches. All
Yeah.
right. And maybe our last live question to ask, but if you all still have questions for Maya, continue to ask them in the chat before we move on, or we'll ask two. Okay. So can you name some beaches in the closer or farther vicinity of San Francisco where you can find bryozoa? So it looks like you have some tags on the map here. I don't know if those are specific beaches.
Yes, so I can drop a link to the chat from where I got this map after I'm done. And so Fort Funston is a great one. Point Reyes National Seashore. There's the Monterey Capitola area. Yeah, but I will put a link to a full map right after this.
Okay, cool. I think we have time for one more question, and then there's a question in the Q&A. I'll have you type the answer to it, Maya, if that's good. Okay, so the one we'll answer live is, do scientists know whether or how bryozoa may have mutated over all that time? So you were talking about there are these fossils from like hundreds of millions of years ago. Yeah, are there any major changes that scientists have seen?
Yes, tons. So basically we started out with all like giant reefs of bryozoa. They only formed really, really big colonies, like coral. And then three kind of just spread to occupy more different roles in the intertidal environment. So now there are some that look like lichen, and they're kind of like little leaflet structures. There's some that look like underwater trees. There are some that just grow flat on rocks. Oh, and there's a special kind that only grows on hermit crabs. And it actually grows in the same spiral shape as the hermit crab shell, and then keeps growing so the hermit crab can just live inside the spiral bryozoan. And then the hermit crab doesn't have to find a new home. So they have that symbiotic relationship. So they've grown to specialize in a lot of ways. There's also one group that can walk, and basically every single individual in the colony has one leg, and then they all coordinate walking around the seafloor. So they do all kinds of weird stuff.
Okay, cool. So with that, let's thank Maya one more time. And Maya, there's just one more question for you in the Q&A box. And so we'll transfer over to our second speaker of the evening.
Our second speaker of the evening is Gardner, who is originally from outside of Cleveland, Ohio. He got his bachelor's degree from Ohio State in chemistry with minors in mathematics and history. He is currently a fourth-year graduate student in the chemistry department at UC Berkeley. Outside of research, he loves walking around the city of Berkeley and rooting on his hometown sports teams. Go Browns! Thank you for volunteering to speak with us, Scott. Take it away.
All right, great. Yes, and yes, go Browns indeed. All right, okay. So I'm really excited to get to talk about my work at the library. This is a really awesome opportunity, so thanks so much for having me here. I want to talk today about what I work on broadly, which is quantum chemistry in service to modern X-ray science, which we'll dive into basically all the confusing bits of this throughout my talk. So just a general outline. I want to start with just generally what got me interested in chemistry. I'll dovetail this into spectroscopy, which is one of my favorite topics. And then from here, we'll go right into X-rays. And I want to talk about how they work and sort of the questions I'm currently asking that I think X-rays can help me solve. Then I'll talk about quantum chemistry, sort of broadly the research I do. And then I want to talk about the sort of computers that I get to use in order to do this research. And then I'll sort of wrap things up at the end. So first things first, what got me interested in chemistry? So something you've probably heard of is sodium. As a metal, sodium alone, when you drop it in water, it makes just a very violent explosion. And I learned about this probably sometime in high school, and explosions are cool. So right away, I'm turned on to chemistry here. There's another element you've probably heard of, which is chlorine, which you might know as the stuff that cleans pools by killing bacteria, or also chlorine on its own is a poisonous gas. And so these are two just really dangerous things. But if you combine them in the right way, and in this case, the green balls are chlorine atoms, and the red ones are the blue ones are sodium atoms, you get sodium chloride, which you probably know as table salt. And so this is just amazing to me that you can take these two just dangerous things, mix them together right, and you get something that we need to live and is honestly quite delicious. So broadly to me, this is what chemistry is. I think it's the study of how atoms are combined and what properties the molecules that we make have. And so sort of pictorially, the way I think about it is we can take our building blocks, which are atoms, and we have all of our atoms are just individual elements off the periodic table, and combine them in different ways to make molecules. And so the periodic table is only so big, we only have 100 or so elements, but we can make just tons of different combinations, and the way we combine them and put them together really changes what they do. And so up at the top here, I have just a molecule I've been thinking a lot about recently. And the way to understand this is all of the balls are individual atoms, and the lines connecting them are individual chemical bonds. And so this sounds like it should be not too bad of a problem, right? We know what all of our building blocks are. We only have 100 of them on the periodic table, and we know how to stick them together to make molecules. But there's a bit of a problem in that these molecules are just really small. And so I refer to this as the problem of size. So on a scale here going from big to small, if we start from something like me size as a human and go down to maybe like a single hair, that's something I can see with my eye pretty well. But any smaller down to cells and bacteria, we really need microscopes to start to see them. And then as things get even smaller, from viruses to DNA to molecules and atoms themselves, we need to go even stronger microscopes, such as electron microscopes. But even those aren't strong enough to see what's going on at the atom level and at the small molecule level. And so this is a big problem if we want to start combining all these building blocks to make molecules, and that we don't even know what the building blocks are because we can't see them. So the way we get around this, and scientists have gotten around this for a very long time, is so-called spectroscopy. And all spectroscopy is the study of how light interacts with matter. And so an example that I think both Isaac Newton and Pink Floyd were big fans of is a prism dispersing light. So you have just some glass triangle. If you shine light in on one side, what comes out the other side is a rainbow of colors. And what's really happening here is the prism is made up of some atoms, some crystals that are just atoms bound together. And when the light hits those atoms, it spreads out into all the different colors. All these different colors, if we look at the entire spectrum of light there is, are just a very, very small slice. And there's others you've probably heard of, such as radio waves or microwaves or infrared radiation. But if we go up higher in energy to something we've probably all heard of, X-rays are just another way we can use light to see what's happening at the atomic level. So X-rays, how do they work? I'm going to start by saying broadly how they work, and then we'll talk about how you might have seen them before. So on one axis here, I have different X-ray energies. So these are just X-rays where I've turned a dial on my X-ray machine and gotten a higher energy X-ray out or a lower energy X-ray out. And what's really cool about X-rays is if I pick the right energy X-ray, I get a peak that comes out that belongs to a specific element. So that might be carbon, it might be iron, which I've highlighted here, or it might be calcium. And so this is kind of a weird idea that different X-rays tell me that there's different elements existing. But if we think about an X-ray you might have had before, it's exactly what we can see is the bones, which are mainly made up of calcium, come out as one color. And then the rest of your hand, which is mainly made up of carbon, comes out as a different color. And so the same idea applies here. The different components of your hand, which are made up of different elements, show up differently in the spectrum. Each individual element leaves a unique fingerprint behind when you shine an X-ray through a material. So that's broadly what I want to say about X-rays. I want to talk about some of the chemistry right now that I'm trying to have X-rays help me understand. And the chemistry that I'm thinking a lot about is, how do molecules fall apart? We talked about how we can build molecules by taking atoms and connecting them together, but what exactly happens if you cut one of those bonds and let the molecule fall apart? So in this case, it's a ring of atoms and they're breaking open. An example of this, why we might care, is sunscreen. You know, if you're out on the beach and wearing sunscreen, you have to reapply it every so often. And part of the reason for that is, as time goes on, these molecules break apart and the sunscreen becomes less effective. So we want to understand, why does that break apart, so that we can go ahead and design better sunscreens that take longer to break apart or don't break apart at all. I think I see a question coming.
We have a question in the chat.
Yeah.
So what is our blood made of? What elements?
Yeah, so that's a great question. So blood is made up of, I think, primarily carbon and oxygen. Blood is mainly, its main purpose is to move oxygen throughout our bodies, but it also has a good amount of iron in it. So what happens is, the oxygen, that building block, binds to the iron, and then as your blood goes through your body, that's what carries the oxygen all around. So that it's very little iron, so it's a little bit harder to see if you get an X-ray done, but if you're clever enough, you could probably see it in the end. Okay, so this is the sort of question I'm thinking about right now, is how do we break molecules that have been formed back down after when we start cutting bonds? And bringing the X-rays back in, what's really cool is, if I have this molecule on the left, and then I break a bond and make it look at this molecule on the right, and then shine X-rays on them, they each leave behind a unique fingerprint. And so if you sort of look at the gray colored in part for both of these, you sort of squint between the one on the left and the one on the right. It's kind of like looking at two fingerprints that they look similar at first, but when you look in at the details, the shapes are different, the numbers on the bottom are different. And so we can think of X-rays as a tool to be able to distinguish, when I study these sorts of processes, if the bond has broken or not. So a bit of a problem with all of this is, if I need an X-ray of my hand, I can sort of flex my hand a little bit and sort of figure out where the bones are, where the fleshy parts are, and sort of have a really good guess of what should come out of the X-ray. And if one of my fingers hurts, maybe I know that bone might be broken. For molecules, it's a lot less clear. How exactly do I go from something that I think looks like this on the left to guessing what a fingerprint would look like on the right? And so the way I go about this is through quantum chemistry. And so quantum mechanics is a really funky topic. Quantum chemistry is something entirely different. I'm going to say it's the application of the principles of quantum mechanics to chemical questions. So using quantum mechanics to try to understand the chemistry. All quantum mechanics is a mathematical theory, which implies energy levels are discrete or quantized. And I really don't like math. It's a really big pain for me. So I like pictures. And there's a picture that we've kind of been seeing all along that is a result of quantum mechanics. So when we draw atoms, which are just electrons orbiting around nucleuses, we always draw these distinct rings, either in circles like the carbon on the left, or just like a couple of distinct rings like the atom on the right. And the reason we draw distinct rings is because of quantum mechanics. That tells us that electrons, they go around the nucleuses, but only in fixed orbits. So the electrons always follow the same paths around and they never deviate, which is a really weird thing, but it's just sort of how the math falls out of quantum mechanics. And with this result, sort of the questions that I usually want to ask are about those orbits of the electrons. I might want to ask, how far should I draw these rings out? Should they be really close to the nucleus? Should they be really far from the nucleus? How many electrons should follow that orbit? Should it be 4, like this outer ring? Should it be 2, like the inner ring? And the question that I personally think a lot about is what is the energy associated with an electron in that inner orbit? As this electron zips around the nucleus, how much energy does it have? And the reason I think about that is because that energy is exactly what this X-ray energy is. So all of these individual X-ray energies are basically the energies of those electrons nearest to the nuclei. So to put all of these pieces back together, this is how we go from, I have a molecule, I sort of know what it looks like, but I don't really know what its fingerprint is. If we go about calculating what is the orbit shape, how big is the orbit, how many electrons are there, we can then use that to sort of figure out what will the fingerprint look like when I shine X-rays on this at the end. So I want to talk a little bit about how I go about doing these calculations. So quantum mechanics is a very mathematical thing. I remember all of my math classes I've ever taken. None of them have ever fallen below the level of very hard. But quantum mechanics, it actually turns out, is mathematically impossible in many situations. We want to know the answer, but it's actually not possible to find it out exactly. And so just like all these classes, when the math was really hard and I didn't want to struggle through it, I pull out my calculator and just start plugging in numbers to try to find out if I can get an answer that way. And nowadays, I'm using a bigger calculator, which is just a computer. So one of the types of calculations I do fairly often is a so-called self-consistent field calculation. And this is really just the guess and check of quantum mechanics. The way it works is I pick some orbits, a red orbit, a blue orbit, a green orbit, and then I'll figure out what the green orbit is. And I'll update all of my orbits, and then I'll figure out what the blue orbit is and update all the orbits, figure out what the red orbit is, update all the orbits, and just continue going around and around and around, figuring these things out until eventually everything agrees with itself. When I update one orbit, none of the other orbits change at all. And that's just self-consistently agreeing in terms of what the orbits should be. I think I see a question popping up.
Yep. What makes quantum physics so hard?
Yeah. So a big piece of why quantum physics is really difficult is because it's mathematically impossible in many situations. It's just a lot of times you can write down an equation for, say, classical physics, so non-quantum mechanics. And a lot of times it's a lot of math and it's very tedious, but in the end you can get to an answer and that answer is mostly correct. In quantum mechanics, that's very often not the case. And it's a really painful thing that it's not. And there's a lot of weirdness that happens in quantum mechanics too. Stuff can be in two places at the same time. And when you measure things, they change. And so it's hard because the math is hard. And it's also really difficult because a lot of it just doesn't make sense. I can't have my water bottle be in both my hands at the same time. It's in one hand or the other. Okay. So these calculations, they have to go around and around and around this loop many, many, many times. Turns out that's a very computationally expensive thing to do. It's like having a million tabs open on your computer and suddenly everything just runs really, really slow because you have to do so many different computations. So in order to be able to do this, I actually need to use a supercomputer. So this picture on the right is a computer that I've been using a lot recently. It's a panoramic picture because all of the green panels you see here are all parts of the computer. This computer is actually as powerful as 1,000 MacBooks combined at once, all working on the same problem. So it's a really, really powerful computer. And a lot of the power that this computer has lies in parallel computation. And so what that basically means is solving for many orbits at the same time. So instead of just going around one loop over and over and over to solve all the orbits, I can take many orbits, work on all of them at the same time on different pieces of this computer, and then collect all of the answers together at the end and make sure it all checks out and then move on. So
we have another question in the chat. Can you play video games on that computer?
I've always wanted to. This is a massive government computer, and every time I sign into it, there's a big warning that says, hey, this is a government computer. No messing around. I've joked many, many times with my friends that I want to load a Minecraft server onto this computer because I think it would be amazing. But unfortunately, I don't think the people who run this computer would be very happy with me doing that. See, I see another question.
Yep, we also have a question. If this is at the Berkeley Science Lab, yeah, can you tell us where this supercomputer in the image is located?
Yes, so I think this one is located up at LBNL, up at Lawrence Berkeley National Lab above the UC Berkeley campus. It actually, I believe, has its own building. You can see this room in the room that it's in is a quite large room. Part of the problem with these computers being so big is they produce a lot of heat. And so a lot of times these computers need their own rooms and their own air conditioning systems just to keep them cool enough to keep them running. Kind of like when your laptop gets hot, it slows down and it's really annoying to use. When this computer gets hot, the same problem. So it needs its own building with its own air conditioning to keep it running.
Is that where you work, Scott?
I work on campus, but it's really cool that I can be on campus and then on my computer here, signed into that computer up there. I can do all of this from remote access. Okay, so I think that's sort of what I'm interested, what questions I'm asking and how I'm asking them. Just sort of to wrap those all up into a few key takeaways, I think. So X-rays, something we're all familiar with, are very useful to doctors and dentists. Turns out they're also very useful to chemists and physicists as well. And pretty much most scientists can find some use of X-rays if you ask them to try hard enough. Atoms and molecules, they can all be identified by their unique fingerprints in the X-ray spectrum. But part of the problem is these are just fingerprints, and fingerprints are always really hard to identify. So we need to do some really difficult quantum chemical calculations in order to be able to match a fingerprint in an X-ray spectrum to a molecule or an atom which it came from. So with that, I want to thank you all for tuning in, and whatever questions you have, I'm happy to answer. Okay.
All right, well, we'll wait just a little longer for questions, but Scott and Maya, thank you so much for these fantastic presentations. I just want to remind all of our viewers that the talks will be available on YouTube soon, shortly after the presentation. And thank you so much for tuning in.
Hey, Scott, I have a question for you.
Mhm.
So when you're, doing your calculations to compare to different fingerprints that come from like experiments, I imagine that in the experiments, they're looking at like a lot of molecules. And I guess, does it matter basically if in an experiment has a lot of the molecules versus one molecule, does that impact your calculations and how you're able to, compare things to?
Yeah, that one unfortunately is a giant it depends. A lot of times, the more stuff I add, the harder the calculation gets. There's just more going on. And so a lot of times, a lot of what I do as a whole is to take a really complicated mathematical thing and say, okay, how many of the hard pieces can I just ignore and say those probably aren't important and just focus on a few little bits here. And so a lot of times when we're doing these calculations, what we start with is, okay, what is the bare minimum we can get away with in order to make a good calculation? And then we see, does that agree with what we saw in the experiment? And if so, great. If not, well, let's add a little bit more on and see if that makes it a little bit better. And let's keep trying this process until eventually we can understand what's happening.
Okay, cool.
I see a question about Sweet Greens. I actually think there's another one of the buildings by Sweet Greens has another one of these very large computers in it. I think. I'm not entirely sure, but I think one of those buildings has the campus version, the UC Berkeley version of the supercomputer.
There's a question. Oh, Oksana, you go for it. I'll answer the thing in the chat.
Yeah, we have a question in the Q&A. Can you work backwards from an X-ray fingerprint to determine the organization of atoms into molecules, or does it only indicate orbital electrons?
Ooh, great question. So X-rays have tons of purpose, tons of uses. One of the probably most useful things X-rays can do is they can tell us protein structures. So if you've ever seen a picture of a protein where there's just things all over the place, a lot of times the way we figure it out, how are those sheets and twists and turns organized, is by using X-rays and turning the proteins into crystals, hitting it with X-rays, letting the X-rays bounce off of it and go in every direction, and then working backwards from every direction the X-rays went to, okay, it must have bounced off of this that way, bounced off of this that way. So that's one way in which you can work backwards from, I've done a scattering experiment where X-rays have scattered off my protein to figure out what actually was that protein in the first place.
I have another question for you, Scott. Is there an exciting discovery or recent development in which quantum mechanics calculations has matched this very difficult, hard to characterize fingerprint recently?
Yeah, so there's been a lot of thought about this recently. So I mentioned it's always the innermost electrons, the ones closest to the nuclei, that we care about. Turns out this is really the only situation those electrons are ever important. For the most part, quantum chemistry, those electrons, they don't really matter that much. And so then there's been a lot of work recently now that people are using these X-rays more and more commonly to say, okay, I know I ignored those electrons before, but now it's time to go in and really give them the fair treatment they deserve to figure out exactly what their X-ray fingerprints are going to end up being.
Okay, thanks.
And Scott, I have one, maybe final if no one else has one in the chat question. What is your favorite molecule?
Oh, that's a tough one.
What do you like to break?
Oh, that's a tough one. That molecule I had on the screen, this little 5-membered ring, I think is my favorite right now because there's some really weird stuff that happens. When it breaks open, you would think it would just break and everything would be fine. But stuff starts going crazy and electrons rearrange themselves in ways I wouldn't expect. So that's my favorite right now, I think, is what I have to go with.
And what kind of atoms are in that ring?
That's a lot of carbons. It's got an oxygen in it, and then a handful of hydrogens as well.
Cool. All right. Well, I think that wraps us up for tonight, it looks like. Thank you guys very much for 2 very different and also super interesting talks. And thank you everyone in the audience for coming.
Thank you, panelists, and thank you, organizers. This has been a fantastic event, and thank you, attendees as well. Next third Tuesday of the month, we'll be hosting Popping the Science Bubble on Zoom. Please tune in, and thank you so much.
Thanks to all the organizers.